Okay, let me first thank the organizing committee for the opportunity to present some of our data. And before I really get into the story and the issue that we are working on, I'd like to take you through at least some introductory slides for those just to refresh the memory on RNAI and antiviral RNAI. Okay, just one slide. I assume many of you will be familiar with Wageningen University and where we are about, but most of you will probably not have been in Wageningen the last couple of years. And just to show you a few pictures of the new campus site that we have moved to during the last 10 years, in which we now are at the moment. So this is more or less the campus site in which most of the buildings of the university have been concentrated. The huge ones are actually the buildings in which we do the teaching towards students, while all the buildings surrounding this site involve the research institutes and many of the laboratories of the university. So just a few nice architectural examples in which the Radix building is actually the building that hosts all the departments related to plant biology and research, in which the virology unit is just here at the edge at the bottom. So as I mentioned, before I really get into antiviral RNAI and some of the work on resistance genes, let me just briefly rememorize the principle of RNAI that you'll probably be familiar with. I have made up a briefly more summarized scheme to simplify a little bit because it is a little bit more complex, but just this would be sufficient for the rest of the talk. So if we are talking about RNAI, RNA interference, RNA sciencing, we basically talk about a mechanism that is induced upon the formation of double-stranded RNA. And these molecules are perceived by Dicer and processed into small interfering RNA molecules, which are approximately 21 to 24 nucleotides in size. From these duplex molecules, one strand gets loaded into a so-called RNA-induced sciencing complex. And this becomes activated with a core element, an argonaute protein, that is next able to perceive RNA target molecules with sequence complementarity to the siRNA strand, and these target molecules will then be arrested during translation or they will be cleaved. And this cleavage basically leads to a degradation of the RNA molecule. Often when people talk about RNAi, they also talk about a sequence-specific RNA degradation mechanism. This mechanism is basically conserved in all eukaryotic organisms, in mammals, plants, nematodes, insects, you name it, where it's involved in gene regulation as well as in development and regulation of chromosome dynamics. And then you talk about euchromatin, heterochromatin. So as said, RNA sciencing is highly sequence specific. If you look in literature though, you will find an enormous load of literature reports on different small non-coding RNA molecules. Maybe it's not readable, but just to give you a snapshot of a long list of small non-coding RNA molecules that really diversify in their size. MicroRNAs, siRNAs, trans-acting siRNAs, piRNAs, and many names. But if it comes to the major pathways, then, and this one summarizes the one in plants, and we have in plants three major pathways. One is the pathway that you will probably also have heard of about microRNAs. And microRNAs basically are processed from host-encoded non-coding genes, which result in a precursor molecule that folds into a stem loop structure, which is imperfect, but then again processed by Dicer into a microRNA molecule, from which one strand then becomes loaded into a RIS complex. And this complex then will mostly lead to a translational arrest of the gene transcripts. And with microRNAs, cells have the ability to regulate the translation, so the gene on, gene off states of gene regulation. Then we have the heterochromatic regions or repeat regions, which refer to, for example, transposable element regions, which are transcribed by Pol4 and amplified by RDR2, leading to double-stranded RNA that is processed by a dicer 3 into 24 nucleotide siRNAs that will load into a RISC complex that then is able to perceive DNA molecules with sequence complementarity to the 24 nucleotide siRNAs. And these sequences will then become methylated. So this is actually the process that one of the previous speakers referred to with the transposable elements, the regulation of the expression and the sizing of transposable elements. And then we have the production of double-stranded RNA involving RDR6 and a cofactor from aberrant, for example, the cleaved RNA products I just mentioned, conversion in double-stranded, the processing by dicer into mostly 21 20 nucleotide siRNAs, and mostly this leads to a RISC complex that will cleave the RNA target molecule. These are basically the major pathways that we distinguish in plants. So although they are all different, in essence, the similarity in action is more or less to all of them the same. It's a gene regulation or it leads to the gene transcript degradation. I can show you more or less similar pictures for insects as well as in mammals, and basically it all is quite similar. If you would look for differences, there are some. For example, as mentioned by one of the previous speakers, in plants we have 4 dicer molecules, dicer 1, 2, 3, 4. Some of these dicers are redundant. If you take dicer, one of the dicers out, it might be compensated by the activity of the others. If you go to insects, you have 2 dicers, dicer 1 and 2. If you go to mammals, you have 1 dicer. So there are differences in the smaller details, but in essence, the whole procedure remains the same. You have the formation of double-stranded RNA, the processing into siRNAs, the loading into RISC, and the perception of target RNA-OD molecules. So if you talk about viruses, and especially plant viruses, we actually are dealing with food, and because we want to guarantee the production of food, not only for the current people, but also for the next generations, which will only become bigger. So we have to be more efficient, but also we have to be aware that we want to do this in a sustainable manner for the future. And if it comes to viruses, as I said, although viruses, as one of the previous speakers mentioned, is only a smaller number, takes only a smaller percentage of the diseases that we face in crops, which is less than 20 or 10%, and fungi and bacteria are much more important, it's still that actually if we talk about emerging infectious diseases, nearly half of them are caused by viruses, not by fungi, not by bacteria. And it's actually only about 20 plant viruses that already by themselves cause an annual crop loss of more than $20 billion, which involves many bulk crops, but food crops like cassava or rice, but also high-value crops like pepper, tomato. So what we are actually doing if we talk about plant viruses is, of course, to get into the biological pathosystem, how the disease is caused, and how for the future can we establish, develop new protection strategies to combat these viruses. And if we talk about defense strategies in plants, then we basically recognize RNAI as a defense mechanism to viruses, as well as resistance genes. And we have different resistant genes if it comes to defense against viruses. So again, the picture of the RNAI principle. So this mechanism can turn antiviral, and specifically against, not only against viruses, but as said, also against the molecular parasites, the transposable elements. They need to be kept silent. So how does this happen? How does this turn antiviral? Actually, not much is changing, as you will see from this slide, because actually what we now start off with is virus that enters a cell, and upon replication, and then you have to consider then mostly we talk about RNA viruses. 90% of all plant viruses are RNA viruses and replicate in the cytoplasm. So when they enter, at some stage during the replication, there will be a double-stranded RNA replicative intermediate formed. Or when transcripts are made, often also RNA secondary folding structures are made. And these are being perceived as double-stranded and will be processed by dicer into the siRNAs. The rest of the scheme is more or less the same. So in this way, RNA turns antiviral because the RISC complex now contains a so-called guide strand of siRNAs that have the origin in viral sequences. So this activated RISC has become antiviral and perceives viral RNA target molecules for translational rest or cleavage. First siRNAs ever discovered were actually improved was in 1999 by the group of David Bolkamp during infections in plants with a PVX virus. It was actually only a couple of years ago, not very long ago, that RNAI was also proven antiviral in mammals. It was only in 2013, and it needed the expertise of two plant biologists, actually two former plant virologists, to prove that RNAI is also antiviral. There was also a long debate until then that many animal virologists never believed in RNAI to be antiviral simply because they were always talking, we already have adaptive immunity. We already have the interferon-induced defense responses. We don't need antiviral RNAI. And it makes some sense, but eventually people demonstrated, and maybe the antiviral RNAI in mammals is not as strong as it is in plants, but it still contributes. The nice thing from all these studies too is that they nicely showed what they call the phase production of siRNAs. Once it starts to cleave, you see it cleaves further on, and you can really see if you clone these siRNAs and you sequence them, you see the phase production nicely. This is something you don't see in plants, because on that point, the RNAi cycle in plants does distinguish a little bit from those in most of the other organisms, which is due to the fact that in plants we also have a so-called amplification cycle. So the amplification cycle is a cycle that basically perceives the aberrant RNAs by host-encoded RNA-dependent RNA polymerases. And it will convert it into double-stranded RNA again, which will be processed by dicer 4 and provides secondary siRNAs. And this is a cycle that keeps on continuing, ongoing. So you get really a large load of siRNAs that will really increase the strength of RNAi. It also means that it is essential to mount a strong RNAi response, also to viruses. Because if we take out the RDRs, we will not have any amplification, no secondary siRNAs, and what we generally see in plants is these plants become highly susceptible to RNA viruses. Of course, viruses need to establish an infection in plants, and they are able to do so. So that means it has to counteract RNAi, because if it doesn't, it will more or less lose the battle. It will become degraded primarily, and maybe even cleared from the host. Viruses, although minimal in genetic load, still encode several different proteins, and there is almost always, any of these proteins that has the ability to interfere. Interfere in the sense that, that it's able to bind long double-stranded RNA and prevents the cleavage into siRNAs. Many bind small interfering RNAs and thereby prevent their loading into RISC. There is even a viral protein that further processes these siRNAs into smaller siRNAs. And these become too small to load and activate RISC. There are also proteins that bind to an activated RISC and prevent the activity of RISC. Or some even bind and target these RISC complexes to the proteasome for degradation. By binding to secondary siRNAs, it also prevents that they move systemically. And systemically means that in plants where Connie Peters earlier talked about systemic acquired resistance, this is also a process that happens with viruses where siRNAs move systemically, will activate risk in systemic tissue before even the virus gets in. So somehow protects that actually the virus will be too virulent in the young leaves and reduce the virulence of the virus systemically. If we talk about the RDRs that are involved in the amplification cycle, then in general it's recognized that there are 3 RDRs involved in the amplification within this cycle, within the RNAI cycle, to target on RNA, which are RDR 1, 2, and 6. This also refers to the 6 RDRs that are recognized in Arabidopsis. So the RDR 3, 4, 5 for long have not been known to have any function in the RNAI cycle. So as said, we have about 90% of the viruses are RNA viruses in plants. There are about 10% are DNA viruses. And the ones that we recognize and are most well known are the Gemini viruses and the viruses. And both are DNA viruses that replicate in the nucleus. Also, these DNA viruses are prone to RNAI, but then prone to 2 different ways of RNAI. One is the pathway that we generally refer to as the post-transcriptional gene silencing pathway. The other one is the transcriptional gene silencing pathway, which involves the processing of double-stranded RNA by dicer tree to generate 24 nucleotide siRNAs. These will then load into a RISC complex that some even refer to as RITS, which stands for RNA-induced transcriptional silencing, loaded with an Argonaut 4 instead of an Argonaut 1 here. And this will be able then to perceive the DNA with sequence complementarity to the 24 nucleotide siRNA sequence and lead to the methylation of the cytosines in the DNA, viral DNA genome sequence. Also, DNA viruses counteract by coding for proteins with the ability to interfere in the RNAi. And the well-known ones, most of them interfere in the methylation cycle, prevent thereby the methylation of the DNA genome, which then still is able to transcribe and eventually replicate. Some also bind the double-stranded RNA or interfere by inhibiting the amplification cycle. So if it comes to the study on viruses and antiviral RNAi, in our lab we mostly focus on 2 viruses. One that I'm not going to talk about today are the viruses, a very interesting group of RNA viruses that is classified in a family with a lot of animal infecting and human infecting viruses. But the other group we are also working on are the Gemini viruses. The Gemini viruses, as well as the viruses, are amongst the most economically important viruses worldwide. So the Gemini viruses are just circular DNA elements. They're only about 2.6 kb in size, Gemini particle, and transmitted by the whitefly Bemisia tabaci, which in Holland, as well as the Gemini viruses, are quarantine viruses and quarantine pests. So we can also justify the work on these viruses because we do not want these viruses in many of our greenhouse cultivations. So if we talk about Gemini viruses and the RNAi, then the way that RNAi is induced by these Gemini viruses is due to the fact that Gemini viruses are recognized by a so-called bidirectional transcription mode. So the left hand and the right hand sided gene transcription from the intergenic region, leading to a kind of overlap in this area. And it's generally assumed, this is not at all proven, but this is the model, that this overlap here gives a template for RDRs and the cofactor to generate a double-stranded molecule that next becomes processed by dicer and leads to antiviral RNA silencing. In the same mode, this template will further on give a continuous, complete double-stranded RNA molecule across all the other overlapping transcripts. And people generally can show that siRNAs are made spanning the entire genome by simply amplifying pieces of the genome like done here, where actually all the genes are separately amplified. You can purify the siRNAs from infected plants, radio-label them, and probe them on this blood, and you'll see that they're all basically produced. These siRNAs at least for sure, contribute to post-transcriptional gene silencing. So this will load into RISC and target viral RNA transcript molecules. But as said, Gemini viruses are also prone to transcriptional gene silencing, which means that somewhere down the line, these double-stranded RNA molecules are also processed by dicer 3 into the 24 nucleotide siRNAs that load into RISC and lead to the corresponding methylation in cytosine residues in the DNA strand. And this has already been nicely shown several years ago by a group in Spain, in which they collected leaves, systemically young leaves, intermediate, and the further down leaves that exhibited different levels of infection. The older leaves were much more severely infected. The intermediate ones with a medium virus titer, the upper ones almost showed a relative rescue of viruses. And when they collected the viruses from these leaves, they also analyzed the methylation profile of the collected viral DNA. And what they observed is that in the severely infected leaves, the methylation of the cytosine residues was relatively low. While if they looked at the younger leaves and collected viral DNA, they saw an increased cytosine methylation level. So clearly, the viral Gemini virus DNA is being methylated and prone to transcriptional gene silencing. So now getting actually to some of the data we have done in relation to a resistance against Gemini virus, because it's difficult to combat Gemini viruses. People in general use insecticides to combat the whitefly because of the limited alternatives. But of course the alternative is to go for breeding against the resistance. And currently there are about 6 resistance genes available for breeding against Gemini viruses. And about 10 years ago we slowly started to try to map and clone these resistance genes. And what we managed several years ago is that we were able to clone and map the TY1 gene, and down the line also showed that the TY3 was an allele from TY1. And we also observed that these genes code for an RNA-dependent RNA polymerase. And now probably, considering the previous slides, a bell will ring within your minds. As mentioned earlier, in Arabidopsis there are 6 RDR genes. They're just listed here, RDR 1, 2, 6, and 3, 4, 5. The 1, 2, 6 are known to play a role in RNAI if it comes to post-transcriptional gene silencing. And with it, the RDR2 involved in the production of the 24 nucleotide siRNAs. But RDR 3 and 4 and 5, no function was assigned at all to them. And it turned out that our TY1 gene is actually a member of this RDR3, 4, 5 class, and not at all these others. So, of course, and easily, you will agree with me that if it comes to the hypothesis, what would this TY1 gene do, you immediately refer and think about this amplification cycle. And so did we. Of course, there's a logical idea behind this, because we know that if we knock down RDR1, 2, 6, we already observed that these plants turn highly susceptible to RNA viruses. There are even situations where people were able to upregulate RDR1, 2, 6, and they observed an enhanced resistance to the RNA viruses, but all to RNA viruses. None of these analyses showed that it increased the resistance or raised susceptibility to the Gemini viruses. So apparently, Gemini viruses are not really strong and prone to post-transcriptional silencing, more likely to transcriptional gene silencing. So our idea was, and that was where our question is TY1 as an RDR1 transcriptionally upregulated, contributing to a strong RNAI response? If so, of course, the next questions, does TY1 contribute to an siRNA amplification and or enhanced genome methylation? The answer to all these questions, yes, yes, yes. Just to show you in summary the data, here we can see the situation in susceptible moneymaker and in the TY1 tomato. And it's clear that in the TY1 tomato lines, the TY1 gene is clearly transcriptionally upregulated. Does that explain the resistance to the Gemini viruses? We don't know, but surely a transcriptional enhanced profile is likely to contribute to a stronger RNAi response. What about the siRNAs? I earlier showed you this picture. You can detect siRNAs produced all over the genome. This is quantified to an internal control, and when we collect the siRNAs from a TY1 resistant tomato land challenged with tomato leaf curl virus, then we do again see the production of siRNAs to all the genes. But clearly, relative to the internal control, we see an enhanced production of siRNAs. Last but not least, what about the methylation? Considering that we actually saw a quite increased amount of CP and C3 siRNAs, which is in this region, relative to what we see here, we looked especially at the methylation profile in this area. So we amplified this PCR by PCR, this region from the viral genome that we collected from these plants, and which we also treated with bisulfite, which converts the methylated cytosine residues, the non-methylated one, in uracils. So by this bisulfite sequencing approach, you can then next estimate which cytosines are methylated or not. And interestingly, if you look at this region, then we actually could distinguish the beginning of the region and the end. And if we looked in the moneymaker, the susceptible line, we saw a certain methylation rate similar in the upper and the further downstream region. But if we collect the viral DNA from the TY1 lines, then we saw actually a similar methylation rate in the upper region. But if we look further down, we saw an increased methylation rate in the viral genome, and which is more schematically diagrammed here. Similar methylation rates in the upper region, so the beginning here, but higher methylation rates here in the TY1 line in the region for the promoter of the coprotein gene, where we observed a stronger increased accumulation of siRNAs. So from this we conclude that with this TY1 we have identified a new resistance gene, a completely new class, because resistance genes mostly come from the class the MBS LRR genes. And this is a resistant gene that does not lead to an HR, does not lead to a complete absence of viral infection. You can still detect a low level of virus titer in there. It's a gene that is apparently transcriptionally upregulated, but likely confers resistance by an enhanced transcriptional gene sizing. And with these findings, we clearly now can assign the RDR3, 4, 5 class at least to play a role in the RNAi amplification. We are still further investigating how and where precisely. But of course, having this resistance gene from the breeder's point and the grower's point of view is, of course, what about the durability of this resistant gene? I mean, it works against tomato yellow leaf curl. Does it work to other viruses? Or what if we mimic a natural situation in which growers often see there is a mixed infection? What would happen if you have a mixed infection with an RNA, another virus, that is also able, somewhere down the line, to interfere in RNAi? Could that compromise the RNAi defense against the Gemini viruses? So, if this is the scheme you're slowly quite familiar with, then a couple of years ago, interestingly, people discovered, that CMV, cucumber necrosis virus, cucumber mosaic virus, which also belongs to one of the most important plant viruses worldwide, encodes for an RNAi suppressor, 2B, we already knew, but this 2B also interferes at the level of AGO4. So it interferes and compromises in transcriptional gene sizing. Why? We don't know. And how? It's likely that 2B has a motive and that's already discovered, what they call a WG motif. And that is a motif that enables proteins to bind to argonaute. So T2B interferes at AGO1 level in the post-transcriptional gene sizing, and also at AGO4, probably by binding. If it's a matter of collateral damage, we don't know. There might be a good reason for, but anyway, it compromises in transcriptional gene sizing. So we asked ourselves, what if you mimic a natural situation and you have a mixed infection of these tomato with TY1, with the Gemini virus and the cucumber mosaic virus? Would CMV interfere here and actually compromise the TY1 resistance? The answer to this question, yes. Here, just look at mostly these numbers. We performed a lot of controls, but this is the essence, where if you challenge TY1 with TY-V, this is the CT value we generally obtain, 28.7. But if we perform a mixed infection on TY1 lines, we generally see almost a 21-fold increase in the TY1, TY-V levels. So, yes, CMV, and it could be any other virus, is able during a mixed infection to compromise TY1. So growers now know that they should not just monitor for TY-V and whiteflies. They also have to be careful to look out for the presence of aphids that might bring in CMV or any other virus. So, of course, we have many other questions that we further want to look into, and these are the last ones. Of course, as I mentioned, we see more siRNAs, but now we are trying to further find out if these are primarily 21 or 24, or both, or maybe specifically only the 24 nucleotide siRNAs. And of course, TY1 is a resistance gene, but not specifically triggered by TY-V. It is triggered, or it works on the principle of double-stranded RNA that processed into 24 nucleotide siRNAs, and not really TY-V specific, but maybe any other Gemini virus. We already did some tests, and we noticed that other big viruses, quite related to TY-V and transmitted by whiteflies, they are also, the TY1 resistance gene also confers resistance to these viruses. But we've also found out that, and not to our surprise, that it also protects against beet curly top virus, a completely different Gemini virus that is transmitted by leafhoppers. And of course, we are interested in, for example, would it confer resistance to cowlimoviruses? Personally, I believe, if I would bet on it, yes. We will investigate it in the near future. RDR345, the TY1 is collected from tomato, and in tomato there is only one RDR45 type of gene. But in Arabidopsis there are 3. So what we also want to find out, the roles of RDR345 individually in the biogenesis of viral or endo-siRNAs and in the epigenetic antiviral defense against Gemini viruses. And at the moment we're also performing TY1 localization studies to further show that this is probably specific for DNA viruses that replicate in the nucleus. So we would suspect that TY1 localizes in the nucleus, and at the moment preliminary data indicates that yes, we localize TY1 in the nucleus, not in the cytoplasm. And that would also explain why TY1 does not confer resistance to RNA viruses that replicate in the cytoplasm. This is the last slide, actually the most important people. I'm just telling their story. Sorry, there we go. I have to go. So, Maarten Verlaan, who spent several years to map and clone the TY1 gene, Patrick Butterbach, a postdoc who did all the analysis on the transcriptional gene sizing, and the work is at the moment being continued by Corien Voorburg, who is working on the siRNA profile, the resistance spectrum, and the localization analysis. With this, I'd like to finish, and I'll be happy to take your questions.